Good morning, folks. Can you guys hear me okay? Yes. All right, welcome back. Happy Tuesday. Let's get started. Guys, any questions, any concerns, any problems, anything at all? Are we doing all right? I'll take your word for it. All right, folks. So today we are going to start a new chapter, chapter 6, and we're moving right along. We are right on schedule, as we should be. And so we're going to start chapter 6 today, and then we're going to finish it on Thursday, okay? And then, of course, I'm going to open the quiz for chapter 6. And chapter 6 can be a little complicated, a little complex, because we're going to be talking about action potential. We're going to be talking about the contraction of a skeletal muscle cell. And the information can be a little overwhelming. So I do want you guys full focus. I do want you to stop me if something is not clear. Okay? Anything that I said went right over your head, just say, Professor, can you explain that again in a different way? And I'm going to do that as many times as I can. Okay? So don't just suffer in silence. If you don't say something, I can't help you otherwise. So let me share my screen with you. All right, guys. So chapter 5 was all about the skeletal system. Chapter 6 is about the muscular tissue. And we all know that we have three different types of muscular tissue, but we're going to focus mainly on this chapter here. We're going to focus on the skeletal muscle only. Okay? We know that we have skeletal, cardiac, and smooth. So we're going to talk about the functions and types of muscle. So we're going to start with the physiology first. We're going to talk about what makes a muscle fiber, what makes a muscle cell, how do muscle fibers move, how they contract, types of muscular movements, and then finally we're going to end this chapter talking about naming of muscles. So we're going to talk about the anatomy. So let's start with the function first. One of the functions of the muscular tissue, and now specifically about the skeletal muscle, is that the skeletal muscle, in combination with our bones, in combination with the ligaments and tendons and joints, is going to allow us to be able to move our body. So no one structure can do that on its own. Okay? A bone without a muscle and a joint and a tendon and a ligament can't do any work. A muscle without a bone, a tendon, ligament, and a joint is not able to move the body. Another function of the muscular tissue is to help to stabilize our body position and maintain our posture. And again, talking about the skeletal muscle in this situation here. Because, folks, we are never fully relaxed. It doesn't matter how deep sleep you are. It doesn't matter if you are in a coma. We're still going to have something called a muscle tone. And a muscle tone, you might have heard of this before, but a muscle tone is a semi-state of contraction of our skeletal muscle cells, okay? To help maintain our body in the position, to help maintain our structure. Our muscular tissue is also very important to store glycogen. And again, talking specifically about the skeletal muscle. What is glycogen, folks? Glycogen is the structure that is going to store glucose. Okay, all the glucose that we ate that we did not use is going to transform into glycogen and is going to be stored in our muscles and our liver. Our muscular tissue also helps to move things out of our body. What do I mean by that? Now I am talking about the smooth muscle within the walls of the digestive tract. So we're going to talk about the digestive system later in the semester. And we're going to see that in the walls of the esophagus, in the walls of the stomach, in the walls of the small and large intestine, we are going to find smooth muscle. What is that smooth muscle doing? The smooth muscle is helping to push the food down the pipe by doing a coordinated contraction that is called a peristaltic contraction or a peristalsis. And throughout the digestive tract, we're going to find a circular muscle, smooth muscle, that helps to control the passage of the food item from one place to another. For example, we have a sphincter between the esophagus and the stomach. We have a sphincter between the stomach and the small intestine. So sphincters are circular muscles, smooth muscle, that they control the passage of food through the digestive tract. And finally, our muscular tissue is also involved with something called thermogenesis. What does that mean, thermogenesis? Genesis means to create. If you had read your Bible, you know what Genesis means. Thermo means heat. So thermogenesis is the generation of heat. And every time that our muscle contracts, they are going to create friction. If you take your hands together and you slide them past each other, you create friction that then generate heat. And it's not a little heat, it's actually a lot of heat because we are about 40% muscles in our body. Okay? So again, let's recap the different types of muscular tissue that we have, their location, the function, do they have striations, or how are they involved with the contraction? So we know that we have skeletal, we have cardiac, and we have smooth muscle. Where do we find each one of them? The skeletal is found attached to the bones, to the skeletal system. The cardiac is going to make the walls of our hearts. And the smooth muscle, they are found in several different places. We find the smooth muscle in our gastrointestinal tract, the digestive tract. We find them in the uterus, in the wall of the uterus, which is going to help to push the big-headed baby out when it's time to deliver a baby. We're going to find smooth muscle in our eye. The iris, which is the colored part of our eye, is made of smooth muscle. We find smooth muscle in the walls of our blood vessels, and they help to control our blood pressure. Okay? Now, we will find striations both in the skeletal and the cardiac. What are these striations? These striations, they are structures that help cell-to-cell communication. And that cell-to-cell communication is something that is very important, very crucial for the function of the skeletal muscle. It is very crucial for the function of the cardiac muscle. But it's not something that is very important when we talk about a smooth muscle. And therefore, they don't have the striations. And we know that only the skeletal is voluntary. The other two are involuntary. Okay, again, all information that we have talked, we have seen it before. All right. So from now on, folks, when you hear me saying a muscle fiber, or when you read muscle fiber, I want you to understand that we are talking about a muscle cell. And that can get students confused sometimes with the nomenclature. Don't be confused. A muscle fiber is a muscle cell. Okay, and we're going to go into a little bit more detail about what we find in a muscle fiber, in a muscle cell. And skeletal muscles, they are attached to the bone in two different ways. We have a connective tissue that looks like a cord, looks like a rope. And then this connective tissue is going to attach the muscle to the long bones, like this bone right here. This connective tissue that attaches the muscle to long bones are what we call the tendons. So tendons are cord-like, rope-like. Now, we also find the connective tissue that is going to attach muscle to the flat bones, like the bones of the skull right here. And those look like a sheet of paper, and they have a name. This is what we call the aponeurosis. So again, what is the difference between a tendon and an aponeurosis? Tendons are cord-like and attach muscles to long bones. Aponeuroses are sheet-like and they attach muscle to flat bones. Okay? Now, if you take a look at the cartoon here, folks, what are we looking at here? We are looking at what we call a muscle bundle. What are muscle bundles? Muscle bundles are the different groups of muscles that we have throughout our body. For example, the biceps brachii is a muscle bundle. The triceps brachii, the deltoids, the pectoralis major, the rectus abdominis, the latissimus dorsi, the trapezius, these are all muscle bundles. And the muscle bundles, they are wrapped by a connective tissue, which we can see here is an extension of the tendon. And this connective tissue is what we call the epimysium. So the epimysium is the connective tissue that surrounds the entire muscle bundle and keep it together. Now, what is a muscle bundle made of? A muscle bundle is made of smaller structures, and we can see these structures here in this dissected muscle bundle. And each one of the structures, these are what we call the fascicles. And one of these fascicles is being pulled out so we can take a closer look at it. And every single fascicle is also wrapped by a connective tissue called a perimysium. So perimysium are the connective tissue that wraps around the fascicles. Now, what are the fascicles made of? The fascicles are made of smaller structures, as we can see right here inside. One of these is being pulled out, and this is what we call the muscle cell, aka the muscle fiber. Okay, and every single one of those muscle cells, they're also wrapped with a connective tissue called the endomysium. So we have epimysium, perimysium, and endomysium, keeping everything together. All right, now what about the muscle cells themselves? Muscle cells, they are made of even smaller little fibers, folks, very, very thin. And these little fibers are what we call the myofibril. And we are going to talk a lot about the myofibrils today. Any questions, guys? Now, muscle cells, again, we are talking about skeletal muscle cells, okay? The skeletal muscle cells, they are quite different from other cells in the body. Remember, when we talked about cells in chapter 3, we talked about one cell having one nucleus. Most of the cells in our body are what we call mononucleated. And that means they have one single nucleus. That's the norm, okay? But some cells in our body are actually multinucleated. And the skeletal muscle cell is one of those cells. Now, how does it become multinucleated? Folks, a skeletal muscle cell, a mature skeletal muscle cell, they are formed from a little stem cells called myoblasts. So myoblasts are little muscle stem cells. And you can see here as a myoblast, these little cells, they have one single nucleus on the inside. Now, what's going to happen is that when they are forming a muscle cell, a skeletal muscle cell, the myoblasts will fuse with one another. And then we're going to have several myoblasts coming in and start to fuse. As they fuse, they are bringing in their own nucleus. So we are going to end up with a skeletal muscle cell that then has several nucleus on the inside. They are then multi-nucleated. Now, as a myoblast, these little guys here, they can undergo cell division. They can go through mitosis. But once a cell becomes a mature skeletal muscle cell, now this cell can no longer undergo cell division. What am I telling you? What I'm telling you is that skeletal muscle cells don't divide. So I have a question for you then. If a skeletal muscle cell does not divide, how do we get bigger? How do we get more muscular when we work out? Folks, what do you guys think? So the question is, how do we get more muscular as we work out?
Is it like you kind of damage the muscle for it to repair itself?
You're on absolutely the right track, Stephanie. So what you're saying is, which is correct, in order for us to grow our muscle mass, we need to cause damage to the skeletal muscle cell. How do we cause damage to them, folks? We work out. We lift heavy weights. Now, what happens when we lift heavy weights? We're going to cause tiny little fractures on the myofibrils. And the damage to the myofibrils, that is going to activate our immune system, right? Because it is a damage, after all, it's a damaged tissue. And the immune system is tightly involved with the repairing damaged tissue in our body. So when we damage the cells, the immune system comes in and is going to start patching up those myofibrils, fixing them. In the process of fixing the myofibrils, the immune system will put in more myofibril inside the cell, making the cell more voluminous. So it's not that one cell becomes two. No, it's that one cell increases in volume. Okay? What are we looking at right here, folks? Here we're looking at a very muscular bull, as we can see right here. And this is, by the way, this is a natural. This is not a genetically engineered bull, okay? This is a type of bull in which they have, and they are, again, born this way, they have silencing of a protein called myostatin. Myostatin. Now, what does that mean? Look, this myostatin gene, which produces a myostatin protein, they are involved with regulating muscle growth. So they don't allow muscle to grow too big. But if you go in and you genetically engineer, now again, the situation here of the bull is natural. He has a silent myostatin gene. But if you look right here in this experiment, guys, this is mice, okay? And what they have done here, they have two different types of mice. The mice here at the top, these guys right here, they are what we call the wild type. What does that mean? A regular old mice. The ones here at the bottom, these are mice that have been genetically engineered to silence the myostatin gene. And when you silence the myostatin gene, the myostatin protein is not gonna be produced. So folks, take a look at the difference of the muscle mass of these two animals right here. This animal right here got the same diet, got the same level of exercise, got exactly the same thing as the wild type mice. The only thing that it has different is that it's not producing the myostatin protein. Now, I have a question for you. Can you think of any possible clinical, medical application of silencing this gene in a patient, in an individual? Can you think how this could benefit someone? What do you guys think? Let's do a little critical thinking here, which is why we're in college, right? The question is, can you think of any application of this research right here in the medical clinical setting?
Maybe disease that causes deterioration?
Of what?
Muscles?
Of muscles, absolutely. There's a lot of several diseases out there which are muscle wasting diseases, and we're going to see one of them at the end of this chapter, not today, on Thursday. But if an individual has a muscle wasting disease, one possibility could be, hey, let's silence the myostatin gene. That can help to reverse the problem here, help to reverse the situation. Might not cure that individual completely, but will certainly slow down the wasting of the muscle. Okay? Any questions, guys? All right. Moving on, let's talk about and let's take a look inside a muscle fiber, inside the muscle cell. And just like any cell in the body, a muscle cell will also have a plasma membrane. However, the plasma membrane of a muscle cell, they have a special name. They're exactly the same as any other plasma membrane, but they are called a sarcolemma. So what is a sarcolemma? Sarcolemma is the plasma membrane of a skeletal muscle cell. What else do we find inside of a muscle cell? We're going to find a lot of nucleus. We have just talked about the fact that the skeletal muscle cell is multinucleated. We're going to find a lot of mitochondria, and that's very important because in order to contract the muscle, we need a lot of ATP. We're going to find a lot of myofibril, and we can see one of the myofibrils right here being pulled out and dissected in the middle so that we can take a closer look inside, which we're going to do next. And then wrapped around the myofibril, we have a special organelle that is only found in a skeletal muscle cell. And that organelle, I want you guys to remember the name, because about 5 minutes from now, I'm going to ask you the name of this organelle. And the name of the organelle is what we call the sarcoplasmic reticulum. What is the function of the sarcoplasmic reticulum? The sarcoplasmic reticulum stores and releases calcium. Again, it stores calcium. And we are going to see what is the role of calcium in muscle contraction. Okay? All right, 5 minutes, I'm going to ask you that question. So let's take a closer look then at the skeletal muscle fiber and see what these myofibrils are all about. Guys, everybody look up here for a second. A skeletal muscle cell is a quite unique cell, even in the way that it looks. A skeletal muscle cell looks like a little tube like this. So if I would finish this cell right here, this is what the cell would look like. Okay, so we're taking a little slice of that muscle cell. And as you can see, inside of this skeletal muscle cell, they are packed with myofibrils. Now, this myofibrils, as we can see right here, let's take a little slice of it and amplify it. The myofibrils, they have pattern to it. Some sections of the myofibril, they are lighter in coloration. Some of the sections of the darker. We can see right here, we have dark, we have lighter, we have dark, we have lighter, we have dark. So what I'm going to do now, I'm going to take this myofibril here and take a picture of it under the microscope. And then we're going to amplify it so we can see a little bit better. But again, we can see the pattern. The light section is called the light band, also called the I band. The dark section is called the dark band, also called the A band. And we're going to see what makes the light band lighter under the microscope, what makes the dark band darker under the microscope. Any questions so far, folks? Is everybody with me? All right. So let's do this now. Let's take this little section of the myofibril here. And I want you guys to take a look right in the middle of the light bands. We can see that in the middle of the light bands, there is this dark line right there in the middle. This dark line is a protein that is called the Z disc. Okay, the Z disc. And when we measure the Z disc of one light band until the neighboring Z disc of the neighboring light band, we get a unit. And that unit between Z disc and Z disc, this is what we call a sarcomere. And a sarcomere is what we call the basic contracting unit of the skeletal muscle cell. Okay, again, the sarcomere is the basic contracting unit of a skeletal muscle cell. And we're going to understand why we call this the basic contracting units. So let's amplify this little section of the myofibril here. And again, identifying some of those regions, the light band, the dark band, Z disc, and then the sarcomere. What I want to do now, I want to take this microscopic picture of the sarcomere, and I want to draw it, make a little cartoon of it, right here at the bottom. And we can line them up here. So we have light band, we have dark band, and again, we have light band. We can see here in the cartoon, folks, that the light band and the dark band are made of different filaments. The light band is made of a thin filament, shown here in yellow. And this thin filament is made of a protein called actin protein. So actin protein makes the thin filament of the light band. The dark band is made of a different filament. They are made of a filament called thick filament, which is made by a protein called the myosin. So myosin makes the thick filament. So now you understand why when we look at a myofibril under the microscope, the light band looks lighter because the light can pass right through the thin filament. So we're going to see it brighter. Now the dark band is going to look darker because the light is going to pass less through the thick filament, which is going to look darker under the microscope. Okay? Now, there's something else that I want you guys to focus here right now, and I need everybody's attention, please. I want you to notice right here, folks, how the thick filaments and the thin filaments, they kind of overlap. There's a little section in which they overlap, right? And the thick filaments, they have these little heads right here, these little structures poking out. These are what we call the myosin heads. And we can see how these myosin heads, they are touching the overlapped section of the thin filament. Imagine my finger here is the myosin head, okay? They are touching the overlapping section of the thin filament. Okay. Now, how does a muscle cell become shorter? We all understand that when we contract a muscle, folks, the entire muscle bundle becomes shorter. When we relax the muscle lengthens. When we contract, the muscle shortens. So another name for muscle contraction is also what we call muscle shortening. So I'm going to try to demonstrate now how does a muscle shorten when we contract the muscle. Okay, and it has everything to do with the sarcomere. Imagine these three fingers right here in my left hand, they are representing the thin filament in the light band. And then we have these two fingers right here on the right hand, they are representing the thick filament of the dark band. When we contract the muscle, guys, what's going to happen is that the thin filament will slide over the thick filament. And it's going to cause the entire sarcomere, the entire myofibril, the entire muscle cell, the entire muscle bundle to shorten as well. Okay, this is what we call the sliding filament theory. And the sliding filament theory says that when a muscle contracts, is due to the sliding of the thin filament over the thick, not the other way around. The thick filament stays in place, but the thin filament slides over it. So it is contraction, relaxation, contraction, relaxation. Okay? Any questions, folks? All right. So let's zoom in here into the filaments, the thin filament and the thick filaments. And again, the thin filament is made of a protein called actin protein. And every single one of these little yellow spheres right here, they are representing an actin protein, making the thin filament, okay? Now, the thick filament is made of a different protein, a protein called myosin. And this myosin protein is quite unique in the way that it looks like here. Everybody look up. The myosin protein has a tail, what we call the myosin tail, and it has a head. In fact, it has two heads right here, what we call the myosin heads. So imagine it's like a golf club with two heads to it, just like that, okay? Now, there's something else that is special about the myosin protein. What's special about the myosin protein, specifically the myosin heads, is that the myosin heads are a type of an enzyme called ATPase. Okay, what do ATPases do? ATPases are enzymes that cleave ATP. When I say cleave, I mean they will grab a phosphate group and they're going to remove it from the ATP, forming ADP plus inorganic phosphate. In the process of removing the phosphate from ATP, what's going to be released, folks? Energy. Energy is going to be released when we break off that phosphate, okay? And that energy is going to be harnessed by the myosin heads. And the myosin heads will do a movement. Everybody look up here, please. This is the movement that the myosin heads are going to do. They're going to move up. And this movement right here, folks, this is what we call the power stroke. The power stroke. And when the myosin heads do the power stroke, what's going to happen? They will grab the thin filament and allow it to slide over the thick filament. So it is the myosin head that grabs the thin filament, allowing for the sliding to happen, allowing for muscle contraction to exist. Okay? How they can do that? They can do that because the myosin heads are ATPases. They cleave ATP, remove a phosphate, release the energy, and use that energy to do the power stroke. Okay? So let's take a closer look then at what's happening when our muscles contract. And for a muscle to contract, we need two very important things. Without them, muscle contraction cannot happen. What are those things? We need ATP and we need calcium. Now, I'm going to talk about what is the role of ATP. I'm going to talk about what is the role of calcium in muscle contraction, which is something that you need to know that I love asking questions about it in the exam. And after I give my explanation, if you still don't understand what does ATP does for muscle contraction, what does calcium do for muscle contraction, you need to ask me questions. Okay? Now, let me show you here, give you a visual aid showing what happens to the entire sarcomere as the muscle cell contract. And what we can see here, we can see in this cartoon, in this microscopic picture, we have three pictures of a muscle cell in different states of contraction. We have a muscle cell that is fully relaxed. We have a muscle cell that is partially contracted. And we have a muscle cell that is fully contracted. Now, I want everybody to look up here and take a look at the sarcomere of the cell in different state of contraction. We can clearly see the sarcomere becoming shorter. Now, what exactly is becoming shorter? Is it the light band or is it the dark band? Well, let's check. If you compare the light band of a partially contracted muscle and a fully contracted muscle, we can again clearly see that the light band is shorter. What about the dark band? Well, let's check right here. No difference. And this is where the sliding filament theory came from. So in the theory, the idea is it is the sliding of the thin filament over the thick, not the other way around. And this is why the light band becomes shorter, but the dark band remains the same. Okay? So let's take a closer look now at the role of ATP and calcium. Everybody look up here, please. What I'm going to do, I'm going to take a little slice of the sarcomere right there. And I'm going to put this under the microscope right here. So what are we looking at? We can see here in blue, this is going to be the thin filament. And then here at the bottom, we're going to see the thick filament. What else can we see? We can see the myosin heads of the thick filament right there, which are interacting with the thin filament. Now, what else do we see? We can also see here in this purple oval looking thing. Folks, everybody look up, please. This is what we call a regulatory protein. What is this regulatory protein doing? This regulatory protein on the thin filament, it prevents the myosin head from binding to the thin filament. Because the myosin head, as I said, it needs to go into a little site called the myosin binding site. And in that way, is going to be able to interact with the thin filament. But that cannot happen if the regulatory protein is on its way. So that means that the muscle cannot contract. The muscle is relaxed. Okay? All right, so let's do this. Let's contract a muscle. In order to contract a muscle, folks, we need to have calcium released into the cytoplasm of the cell. And here comes the question that I told you about maybe 10 minutes ago. What is the name of the organelle that stores and releases calcium in the skeletal muscle cell?
The sarcoplasmic reticulum.
Absolutely, the sarcoplasmic reticulum. So now, in order for us to contract a skeletal muscle, we need to send a signal to the skeletal muscle cell telling the sarcoplasmic reticulum to release the calcium into the cytoplasm. And we can see these little red spheres right here, guys. These are the calcium. They're representing the calcium. What is the calcium is going to do? The calcium is going to bind to the regulatory protein, as we see right here in the little cartoon. And once the calcium binds to the regulatory protein, it will remove the regulatory protein out of the way. Allowing what to happen? Allowing the myosin head to stick the head in the myosin head binding site and interact with the thin filament. What comes next? Next, the myosin heads, which are ATPases, they're going to cleave ATP, release the energy, and harness the energy to do what, guys? To do the power stroke. And when the myosin heads do the power stroke, they allow for the sliding of the thin filament over the thick, leading to a muscle contraction. Guys, any questions? All right, can anybody tell me what is the role of ATP in muscle contraction? If you don't have any questions, you can answer that. So the role of ATP in muscle contraction. Which is a great exam question. Help me out. Come on, guys. I just asked if you had any questions. Nobody has any questions. So tell me, what is the role of ATP in muscle contraction? How does ATP allows for muscle contraction to happen? You can explain in your own words. That's fine.
Provides energy?
I need a little bit more than that. You need, so that wouldn't cut in the exam. I need a little bit more information. It is the interaction with the actin and myosin. No, it's not the interaction with the actin and the myosin.
It helps to re-energize the mitosin.
Re-energize. Well, let me ask this again. Does anybody have any questions? And this is what I talk about every single time. You guys just let the stuff go by. Nobody stops me. Nobody asks questions. Why? If it's clear that you don't fully grasp what's... Okay, let me ask then calcium. Let's try another one. Can anybody tell me what is the role of calcium in muscle contraction? It's like pulling teeth with you guys. So does anybody, okay, your turn then. Does anybody have any questions? I don't understand. I'm at a loss here. Nobody have any questions, but nobody can answer the questions that I ask you. How does that work? All right, no questions. Moving on. All right, guys, so let's watch this little video here that kind of summarizes what we've been discussing so far in terms of the sliding filament theory and the role of ATP and the role of calcium in muscle contraction. Maybe now it's going to be a little bit more clear on your end. You share here.
Movado is synonymous with modern design, inspired by the sun at high noon.
Muscle contraction is at the basis of all skeletal movements. Skeletal muscles are composed of muscle fibers, which in turn are made of repetitive functional units called sarcomeres. Each sarcomere contains many parallel overlapping thin, actin, and thick myosin filaments. The muscle contracts when these filaments slide past each other, resulting in a shortening of the sarcomere and thus the muscle. This is known as the sliding filament theory. Cross-bridge cycling forms the molecular basis for this sliding movement. Muscle contraction is initiated when muscle fibers are stimulated by a nerve impulse and calcium ions are released. The troponin units on the actin myofilaments are bound by calcium ions.
This troponin unit is the regulatory protein I was talking about earlier.
The binding displaces tropomyosin along the myofilaments, which in turn exposes the myosin binding sites. At this stage, the head of each myosin unit is bound to an ADP and a phosphate molecule remaining from the previous muscular contraction. The myosin heads release these phosphates and bind to the actin myofilaments via the newly exposed myosin binding sites. The two myofilaments glide past one another, propelled by a head-first movement of the myosin units, powered by the chemical energy stored in their heads. As the units move, they release the ADP molecules bound to their heads. The gliding motion is halted when ATP molecules bind to the myosin heads, thus severing the bonds between myosin and actin. The ATP molecules are now decomposed into ADP and phosphate, with the energy released by this reaction stored in the myosin heads, ready to be used in the next cycle of movement. The myosin heads resume their starting positions along the actin myofilament and can now begin a new sequence of actin binding. The presence of further calcium ions will trigger a new cycle.
All right, guys, any questions? Okay. So far we have zoomed into the skeletal muscle cell, right? And we looked at the molecular level, what's happening when a muscle cell contracts. But we have to remember that we are talking about skeletal muscle cells. And skeletal muscle cells are what? They are voluntary, meaning that we control them. We tell the muscle to contract when we want to, which is different from the other two, this cardiac and the smooth muscle. So what we need to do now then is to understand what is that connection between the brain and the muscle, the skeletal muscle cell. So how does the information flow? What is that information? So we need to put the whole picture together right now. So things are going to get more complex. And this is why I really wanted us to focus on, at the molecular level, the sliding filament theory, understanding ATP, understanding calcium, so that as we add the more information, things get a little bit more clear instead of making things more complicated. Okay? So what we need to talk about right now, we're going to talk about what we call the neuromuscular junction. And I love asking questions about this in the exam. You're definitely going to see questions about this in the exam. What is the neuromuscular junction? The neuromuscular junction, as the name implies, is a junction, is a union between a neuron and a skeletal muscle cell. Is the point where they are going to join each other. okay, is the point of junction between a neuron and a skeletal muscle fiber, skeletal muscle cell. And I want everybody to look down here in this microscopic picture. All right, and this is a very high magnification. We're talking about 1,650 times magnification in order for us to be able to see this. What are we looking at right here? This long tubular things right here, these are the skeletal muscle cells right here. So they're the skeletal muscle cells. And we can see right here in this lighter coloration structure, this is a motor neuron. What is a motor neuron? A motor neuron is a neuron that is attached to the skeletal muscle cell. And we can see that this motor neuron, they have little extensions. Imagine my hands, okay? They look like a motor neuron. My fingers here, these are like axon terminals of a motor neuron. So this is the end of the neuron. What does this axon terminal do? The axon terminals, guys, they hover over a muscle cell and they're going to touch that muscle cell. Again, look at my hand right here. My hands, my fingers are the axon terminal. My arm right here is a skeletal muscle cell. This motor neuron is going to come here and hover over this skeletal muscle cell. The point where I'm going to have an axon terminal, which is going to hover over the skeletal muscle cell, this is what we call a neuromuscular junction. Okay? And what we're going to do right now, we're going to take a closer look at the neuromuscular junction and see what is happening right at that junction. What is going on right there that allows this motor neuron to control the contraction of a skeletal muscle cell? So we're going to talk about what we call the events at the NMJ. And NMJ stands for neuromuscular junction. Everybody look up here, please. I will ask you questions about this in our next exam. I'm going to use a figure just like this one's right here in the exam. And you need to be able to identify these different structures that we're going to talk about right now. Okay, so what are we looking at down here? Everybody look up here. Can you see this little rectangle? So this little rectangle, we're going to amplify it and blow it up so we can take a closer look at it. So we are looking at the neuromuscular junction right here. And I want you guys to notice something. Here in yellow, we have the axon terminal of the motor neuron. Right down here, we have the sarcolemma of the skeletal muscle cell. Okay, again, this is the axon terminal hovering over the skeletal muscle cell. And what I want you guys to notice is that in between the axon terminal of the motor neuron and the sarcolemma of the skeletal muscle cell, there is a space right here, folks. I am filling in this space right here. This space is a very important space, and it does have a name. This space right here is what we call the synaptic cleft, also called the synapse. So what is the synapse? What is the synaptic cleft? The synaptic cleft is the space between the axon terminal of the motor neuron and the sarcolemma of the skeletal muscle cell. Now, when we want to contract a muscle, we are going to send a signal from our brain all the way to that muscle. Everybody look up here. Imagine I want to contract the muscle of my finger, okay? I'm going to send a thought, a signal from my brain, and that thought, that signal is going to travel from neuron to neuron, down my neck, and up my arm until the signal gets to the muscle that controls my finger. What is that signal? Guys, that signal is an electrical impulse that we call action potential. You absolutely need that you understand what action potential is. And if you look right here in the cartoon, and we can see these arrows right here, these little purple arrows, they are representing the action potential traveling through the neuron. And we can see that eventually this action potential will get to the axon terminal. And we can see the action potential right here, the arrows. And now what's going to happen once the action potential gets to the axon terminal? Once the action potential gets to the axon terminal, is going to induce the release of little chemicals called neurotransmitters. So we can see these little green spheres right here, and I hope everybody's paying attention because this is important. And we can see that these little green spheres right here, inside these little vesicles, once the action potential arrive at the axon terminal, they are going to induce the release of these little chemicals into the synapse. Now, what's going to happen to these little neurotransmitters with these little chemicals? Guys, there are several different types of neurotransmitters, which again are chemicals. Each one of these neurotransmitters, they play a different function in different parts of the body. The most important neurotransmitter for skeletal muscle contraction is a neurotransmitter called acetylcholine. You have to remember the name of acetylcholine. So we are looking at right here in green, these little green spheres in the synaptic cleft, they are the acetylcholine. What is the acetylcholine is going to do, guys? The acetylcholine is going to bind to little receptors on transmembrane proteins. And these transmembrane proteins are embedded in the sarcolemma of the skeletal muscle cell. These transmembrane proteins here, they are ion channels. They are ion channels. What do they mean? They allow sodium to pass right through it. So because sodium is charged, right? Right here, it has a positive charge to it. Sodium cannot cross the phospholipid bilayer through simple diffusion. Sodium can only go in and out of the cell through a transmembrane protein that we call an ion channel. Now, usually, when a skeletal muscle cell is relaxed, the ion channels are closed, folks. They're shut down. What does that mean if they are shut down? Which means is that there's a lot of sodium outside of the cell right here, and the sodium cannot enter the cell. The sodium is going to stay right there outside of the cell. Okay, so this is out of the cell. And then on the other side, this is inside of the cell. So I'm going to repeat that. When the skeletal muscle cell is relaxed, the ion channels are closed, which means that the sodium starts to accumulate outside of the cell. Now, once the neurotransmitter, once the acetylcholine enters the synaptic cleft, the acetylcholine is going to bind to the ion channel and will allow the ion channel to open. Guys, the acetylcholine is like a key to open a door. What is the acetylcholine opening? What door is it opening? It is opening the door for the sodium to enter the cell. Once the ion channels now are open, because of the acetylcholine binding to it, now we're going to have a flood of sodium going inside of the cell, moving by diffusion. More specifically, facilitated diffusion, moving from high concentration outside of the cell to lower concentration inside of the cell. Okay, now once all the sodium enters inside of the cell, folks, the polarity across the sarcolemma will change. Before the sodium comes in, inside of the cell was negative, while outside of the cell was positive because of all the sodium outside of the cell. But as soon as the ion channel opens, now and all the sodium enters the cell, we're going to have a reverse of the polarity across the cell membrane. When the sodium goes in, inside of the cell becomes slightly positive, and outside of the cell becomes slightly negative. And that change in membrane polarity, guys, that leads to an electrical impulse called action potential. What does the action potential do for a skeletal muscle cell? The action potential will trigger the sarcoplasmic reticulum to release calcium. And we know the rest of the story. Once the calcium enters the cell, the skeletal muscle cell, in the cytoplasm of the cell, the calcium is going to bind to the regulatory protein in the thin filament, remove it out of the way, allowing the myosin head to stick to the myosin binding site. Once the myosin head sticks to the myosin binding site, the myosin heads are going to cleave ATP, release the energy from ATP, and harness that energy to do a power stroke. And when the myosin heads do the power stroke, we have the sliding of the thin filament over the thick, leading to a muscle contraction. Now, what if we want to relax this muscle here? What do we need to do first? The first thing that we need to do in order to relax a muscle is to remove the acetylcholine from the synapse. Because as long as the acetylcholine is in the synapse, the ion channels will be open. Sodium will enter the cell. Action potential will continue to be generated. But as soon as we remove the neurotransmitter, the acetylcholine from the synapse, now the ion channels once again are going to close. And if the ion channels are closed, the sodium is going to stop going inside of the cell. And if the sodium stops going inside of the cell, no action potential will be generated. And if there is no action potential, there is no release of calcium inside the skeletal muscle cell. So now the skeletal muscle cell cannot contract. Okay. Any questions, folks?
Yeah, I had a question real fast.
Please do.
Can you real fast clarify? So action potential, does that mean it stays action potential from the moment like the thought to move your fingers like from your brain and then from the sodium going into the muscle, it just stays action potential the entire time?
That's a great question. So we're going to elaborate this a little bit more. But the first thing I want you guys to understand is that action potential is electricity. It's an electrical impulse, okay? And the function, the job of this action potential, because what happens is you can imagine that between our brain and my finger right here is not one single neuron, right? That is going to extend all the way from my brain to my finger. It's going to be hundreds, if not thousands of neurons connected to one another, sending the message along. What the action potential does is that when the action potential travels through the neuron and gets to the axon terminal, the action potential allows for the continuation, the propagation of the action potential to the new neuron until the action potential gets to the skeletal muscle cell. Because when the action potential finally gets to the axon terminal of the motor neuron, now the action potential is going to induce the release of neurotransmitters into the synaptic cleft. So to answer your question, yes, it is the action potential, it is the electrical impulse that keeps on passing from neuron to neuron to neuron. So what we call a thought is an action potential. Okay? Good question. Any other questions, guys? All right, and we're going to elaborate on this a little bit more as we go along. So let's talk more then about the action potential. Because again, this is something that will absolutely be on the exam. You need to master it. You need to understand the basic concept here, the basic idea that I'm presenting. Okay, first of all, what is an action potential? An action potential is an electrical impulse. And we can measure action potential inside a cell. There are little devices in which we can put a little probe inside of the cell, like we're looking at right here in this cartoon, guys. We can put a little probe inside of the cell and measure the action potential, measure the electrical impulse. Very important. When the skeletal muscle cell is at rest, meaning relaxed, the polarity inside of the cell is negative. We can see right here is negatively charged inside of the cell, while outside of the cell is positively charged. This is what we call the resting membrane potential. What creates this resting membrane potential? The accumulation of sodium outside of the cell, and remember, sodium is positively charged. So all of the sodium outside of the cell that wants to come inside of the cell to balance things out, but it can't because the ion channels are closed. So this is what we call the resting membrane potential. Now, once we have an action potential arriving at the NMJ, the neuromuscular junction, that action potential will induce the release of acetylcholine into the synapse. Okay? Once the acetylcholine goes into the synapse between a motor neuron and a skeletal muscle cell, now the acetylcholine will bind to the ion channel, opening the ion channels up. Once the ion channels are open, we're going to have a flood of sodium going inside of the cell. Once the sodium enters the cell, what is going to happen to the polarity of the skeletal muscle cell across the membrane? Inside of the cell is going to go from negative to positive, and outside of the cell will go from positive to negative in a split second. And it is this change, this very abrupt, this very fast change in membrane polarity from negative to positive and positive to negative, that creates a spark. And that spark, that electrical activity, is what we call the action potential. Now, we need to understand what the action potential does for the skeletal muscle cell. And the answer is the action potential, once it has been created, will induce the sarcoplasmic reticulum to release calcium. And once again, once the calcium is released, we know the rest of the story, because that then eventually will lead to a muscle contraction. Okay? Guys, are we clear on this? Anything that is not clear, still iffy? So what is this graph right here? Everybody look up for a second. These two graphs right here, guys, they are representing two different things, okay? The one on top is representing action potential inside the skeletal muscle cell. The one at the bottom is representing the tension on the muscle of the skeletal muscle cell, the contraction. Everybody look up here. When the cell is at rest, inside the skeletal muscle cell is minus 85 millivolts. We have seen here, it is negative, right? Now, let's open the ion channels and allow the sodium to enter. And let's track it right here with the action potential inside the skeletal muscle. The action potential goes from minus 85 all the way to plus 30 millivolts. What is that telling us, guys? It is telling us that inside the skeletal muscle is going from negative, minus 85, to positive, plus 30 millivolts. What's going to happen to the tension in the muscle cell? Look right here. It's going to go from 0, when the ion channels were closed and the cell was at rest, all the way up. And then after that is done, going to go back down. So we're going to have contraction, and then it's going to relax again. Okay, so you need to understand, in order for a muscle to contract, we need to have action potential first. There is no muscle contraction without action potential. All right. Now I have a question for you guys. We understand that the acetylcholine, once it is in the synapse, it will keep the ion channels open, right? Allowing then for the sodium to come inside of the cell, action potential to be generated for the muscle to contract. But then, right after the acetylcholine goes into the synapse, the acetylcholine is going to be removed from the synapse by an enzyme called acetylcholine esterase. So what is acetylcholine esterase? Acetylcholine esterase is the enzyme that removes acetylcholine from the synapse, allowing the muscle cell to relax. Okay? That's what it does. Now, during the war, World War II, the Germans were very, very good scientists, very good chemists, and they were very good at designing ways to kill people the most effective and the most brutal way, and the greatest amount of people. That was Nazi Germany. And the chemists, they came up with a man-made gas that they created called the sarin gas. What does the sarin gas do? Sarin gas is a very effective way to kill people. Thousands, if not millions of Jews died in concentration camps due to inhaling sarin gas. What does sarin gas do to the body? Sarin gas inhibits acetylcholine esterase. You tell me what's going to happen to an individual if they breathe in a sarin gas. How do they die, folks? Again, you understand the mechanism here. Sarin gas inhibits acetylcholine esterase, which is the enzyme that removes acetylcholine from the synapse. How can someone die when they're exposed to sarin gas? Let me ask you this in a different way then. Can your muscle, skeletal muscle, relax if you are exposed to sarin gas?
Yes.
Can you relax? It's not that the muscles aren't contracting. It's actually the opposite. So let's understand this here once again. Acetylcholine esterase is an enzyme that removes acetylcholine from the synapse. Once acetylcholine is removed from the synapse, then the muscle can relax, right? Because as long as the acetylcholine is in the synapse, the ion channels will be open, sodium will continue entering the cell, action potential will continue being generated. The muscle will continue to contract. Now, if you inhibit the acetylcholine esterase, that means the acetylcholine stays in the synapse, which means that the muscle stays contracted. Now, you might be thinking to yourself, okay, how does that kill someone? Guys, what is the name of the muscle that we have right under our lungs that helps with the inhalation and exhalation? The diaphragm. When we contract the diaphragm, we inhale. When we relax the diaphragm, we exhale. So if you are exposed to sarin gas, you're going to be able to inhale, no problem, because you can contract your muscle. Are you going to be able to exhale? No, because you can't relax the muscle. Guys, is this clear? Do you understand what acetylcholine esterase does? Any questions? All right. So let's watch another little video here. And this video, once again, is going to summarize what is happening at the NMJ, at the neuromuscular junction.
Skeletal muscle contracts only when stimulated by nerve impulses from a motor neuron. Without innervation, muscles become paralyzed and eventually atrophied. The axon of a motor neuron usually gives out many branches, supplying multiple muscle fibers. These fibers contract in unison when activated and constitute a motor unit. A motor unit can contain anywhere from just a few muscle fibers to thousands of them. Small motor units are found in muscles that require finer control, for example, muscles that are responsible for subtle movements of the eyes. Large motor units are found in larger muscles that require strength, such as muscles of the arms and legs. Muscle fibers of the same motor unit are usually not clustered together, but scattered throughout the muscle. This way, stimulation of a motor unit has a slight but uniform effect on the whole muscle, and not causing a small, localized twitch. The strength of a muscle contraction is determined by the number of motor units that are activated at one time. Even at rest, most muscles are in a partial contraction state called muscle tonus, which is maintained by alternating activation of a small number of motor units. The connection between a motor neuron and a muscle fiber is called a neuromuscular junction, which is basically a chemical synapse between the nerve terminal and a specialized area of muscle cell membrane called the motor end plate. When an action potential reaches the nerve terminal, it causes the release of the neurotransmitter acetylcholine into the synaptic space. Acetylcholine then binds to nicotinic receptors on the end plate. Nicotinic receptors are ligand-gated ion channels. Upon binding to acetylcholine, they open to allow sodium to enter the cells, depolarizing the cell membrane, producing the so-called end plate potential. An action potential is generated in the muscle cell only when the end plate potential reaches the threshold required to activate voltage-gated sodium channels located outside the end plate in the neighboring membrane. When activated, these channels allow faster influx of sodium, further depolarizing and eventually reversing the polarity of the cell membrane. At this point, voltage-gated potassium channels open for potassium to move out, quickly returning membrane voltage to its original resting value. Once generated, the action potential spreads like a wave, thanks to similar voltage-gated ion channels located throughout the muscle fiber. The action potential also runs deep into the fiber via T-tubules to reach the sarcoplasmic reticulum. Here it activates voltage-gated calcium channels, releasing calcium from the sarcoplasmic reticulum into the cytosol of muscle cells. Calcium then sets off muscle contraction by the sliding filament mechanism. This mechanism is described in another video. Another important component of the neuromuscular junction is the enzyme acetylcholinesterase. This enzyme removes all acetylcholine molecules that do not immediately bind with the receptor and those that are done activating a receptor. The enzyme action essentially terminates synaptic activation, giving the muscle time to relax and thus preventing continuous contraction that would result in muscle spasms. Substances that cause muscle weakness or paralysis do so by interfering with the function of neuromuscular junction. Botulinum toxin prevents acetylcholine release from the presynaptic side of the junction. Some other toxins attach to nicotinic receptors, blocking acetylcholine from binding but do not open the ion channel. Certain drugs lodge into the channel of nicotinic receptor, blocking the passage of sodium. All these substances prevent activation of muscle cells and cause flaccid paralysis. On the other hand, some pesticides inhibit acetylcholinesterase, preventing degradation of acetylcholine, causing continuous activation of muscles. That's how they induce muscle spasms and cause spastic paralysis.
All right, guys, any questions? All right, moving on. Now let me talk a little bit about what we call a myogram. What is a myogram? A myogram is a device, like a test that we can do, in which we can actually record the action potential and the muscular contraction of a muscle. So let's say someone is concerned about muscular wasting disease, okay? And then they're going to go see the doctor, they're going to go see the specialist, and they're going to check what is the strength of the muscle contraction. Not only they're going to look at the tension of the muscle, but they're also going to look at the generation of action potential, which is the electrical impulse. All right. Now, you don't really need to memorize all the stuff right here, but I want you to understand the most important thing that you need to get from this is to understand that the signal, the action potential, is required in order to activate muscle contraction, because there is no muscle contraction without action potential. Okay, and action potential happens first, then muscle contraction is going to take place. That is the only thing that you need to get from this slide right here. Now, in this slide, I will absolutely ask you questions about this in our next exam. Guys, I'm going to use the same figure that we have right here on the screen right now. What else can I tell you? You need to understand this. You need to be able to remember this. What are we going to talk about right now? We're going to talk about how our body adjusts the use of energy depending on the type of activity that we are doing. Our body is smart like that. Our body works in an autonomic ways, folks. We don't have to be thinking about that all the time. Our body does that on its own. Okay, so let's look at where this energy comes from and how our body adjusts to different activities, to different level of activities. All right. When we start to exercise, let's say you start to run right now, a short run. As soon as you start to run, where will our skeletal muscle cells get the energy that it needs to keep contracting the muscle? Well, we have a reserve of ATP inside the skeletal muscle cell. Every cell in our body has a certain amount of ATP already in there. I want to make an analogy here. Imagine that you go out to the supermarket and you want to buy something, but you only have cash. You don't have any credit card. So you can only buy whatever is inside of your wallet. So at the beginning of an exercise, the skeletal muscle cells will use the ATP that is already in there, has already been made. Okay? So it will take ATP, break it down into ADP plus inorganic phosphate, generate energy that way. Now, what if you continue to exercise? If you continue to exercise, now the ATP in your skeletal muscle cell is gone. You use it all. There's nothing left. So now what your skeletal muscle cell is going to do is that they're going to break down a compound, a chemical in the skeletal muscle cells, and the chemical is called creatine phosphate. You might have heard of creatine monophosphate before. It is a supplement that people take to help them work out, to get more out of their workout. What does creatine phosphate do? Creatine phosphate is going to donate a phosphate to ADP. So when we add a phosphate to ADP, what do we get, guys? We get ATP. So again, now the cell can use that ATP and keep on contracting as we continue to exercise. Now, as you can imagine, again, the creatine phosphate will eventually run out of the skeletal muscle cell. What is our body going to do now for energy? Now, as we continue to exercise, the cell needs a fast, dirty way to get ATP. And it's going to do that through what we call glucose fermentation. The skeletal muscle cell is going to take the glucose that we ate for breakfast and is going to send through a pathway called fermentation. And a fermentation of glucose is not a very effective pathway, but it's a quick, fast, dirty, easy way to get some ATP out of the glucose. Because fermentation is an anaerobic process. It does not involve the oxygen gas. But out of one glucose, I can only get 2 ATPs from fermentation. Again, not very effective. But if you stop exercising right then, you're going to be fine. It was fine using the glucose for fermentation. It served the purpose. What if, though, instead of running a short run, you are going to run a marathon? Now your body will adjust to that level of activity. And instead of taking the glucose and sending the glucose through fermentation, the skeletal muscle cells are going to take the glucose and send it through glycolysis, which is, again, it's a different pathway. And glycolysis, different from fermentation, is an aerobic process. It does require oxygen gas in order to be able to generate ATP from glucose. But look at this, guys. From one glucose, we can get up to 34 ATPs. Much, much more efficient than if I take the glucose and send it through the fermentation pathway. Now, at the end of the day, the energy source for us, for everything in our body for that matter, is ATP. The question is, how are we going to get that ATP from? Are we going to get the ATP from the ATP already in the cell? Are we going to get it by generating ATP from creatine phosphate? Are we going to get ATP from glucose fermentation? Or are we going to get ATP from glucose sent through glycolysis? Okay? Any questions, folks? All right. Moving on, just a couple more slides here, and then we're going to call it a day. I know it's been a long morning so far, but let's talk about the different types of muscle contraction, okay? And we can divide muscle contraction into 2 groups, and you might be familiar with those terms. We have what we call the isotonic contraction, and we have what is called an isometric contraction. Within the isotonic, it can be concentric or eccentric. And what is the difference between the 2? Again, you might have heard it before, but you don't fully understand what they do. How are they different? So let's understand that once and for all. Remind me again, and this is from tonicity. Remind me again, what does iso means, guys? What does iso means?
It means the same.
It means the same, absolutely. So let's look at the words right here. Isotonic. Isotonic means we're going to have the same tension in the muscle during the contraction. The tension is not going to change. Now, what about isometric? Well, isometric means we're going to have the same length of the muscle during the contraction. Let's understand the difference between the two. In an isotonic concentric contraction, okay? In a concentric contraction, let's say that you have a dumbbell in your arm. All right, I have a dumbbell in my arm right here. When I bring the dumbbell up, do I need the tension? Do I have tension in my muscle, in my biceps? Absolutely. Now, what is happening to the length of my biceps as I bring the dumbbell up? Is it staying the same or is it changing, guys? It's changing. It's becoming shorter, right? We know that when we contract the muscle becomes shorter. So this here is an isotonic because the tension in the muscle doesn't change, but the length of the muscle does change. And because now my muscle is becoming shorter, this is what we call a concentric isotonic contraction. Now let's do the opposite movement right here. If I take the dumbbell and I bring it down, do I have tension in my muscle? Yes. Does the tension stays the same? Yes. What about the length of the muscle? The length of the muscle is changing because now it is lengthening. And when we lengthen a muscle under tension, we call this an eccentric contraction. Now, what about the isometric? In an isometric, let's say that I take a dumbbell, okay? And I hold the dumbbell in place. I'm not bringing it down. I'm not bringing it up. Hold it in place. Is there tension in the muscle, guys? Yes, absolutely. Is the length of the muscle changing? No, because it is staying in place. So this is what we call this. When we hold the dumbbell, when we hold the weight in place without upward or downward movement, we are doing a isometric contraction. Okay. What is the best type of contraction to gain muscle mass? There are studies out there that have shown that the most effective muscle contraction to gain muscle mass, because it causes the most damage to the muscle, is eccentric isotonic contraction, when the muscle is lengthening. And that is because that creates more tears to the myofibril when we have tension in the muscle as it lengthens. Okay. Now, will our muscle become fatigued after we exercise for a while? Absolutely. What does fatigue mean, folks? Well, let's look at the molecular level. Fatigue means that the level of calcium in your skeletal muscle cells starts to go down. Again, if you don't have enough calcium, you can't contract the muscle. Fatigue also means that the level of ions like sodium is going down. Because if the sodium doesn't enter the skeletal muscle cell, we can't have action potential generated. Muscle fatigue means that the ATP in your skeletal muscle cell is going down. And if you don't have ATP, your myosin heads cannot do the power stroke. That's what a muscle fatigue means, a decrease in calcium ions and ATP. Now, what happens when we go into oxygen deficit as we continue to exercise, right? Your amount of oxygen goes down to your cells because you're breathing in hard. They're consuming more oxygen. And what's going to happen is that now all the glucose is going to go through fermentation. Because remember, in order to have glycolysis, we need the oxygen gas. Glycolysis only works if it is under aerobic condition. Because if you don't have oxygen, enough oxygen gas, if you are oxygen deficit, now we can only get ATP by sending the glucose through fermentation, which will give a couple of ATPs. Now, not enough to sustain the activity. Okay. Another problem with fermentation is that fermentation leads to the formation of lactic acid, an accumulation of lactic acid in the muscle. This is why your muscle hurts after you exercise, because of the accumulation of lactic acid in your muscles as your skeletal muscle cell uses fermentation. Okay. Is it true, guys, that if you don't use your muscle, you will lose it? What do you guys think? Absolutely resounding strong yes. I mean, we can look at those guys, Arnold Schwarzenegger, Sylvester Stallone, all those bodybuilders. They used to have huge muscle. By the way, this got to be Photoshop. I don't think that's real. But anyway, they had these huge muscles, and then you stop exercising, your body will get rid of the muscle. I have a question for you. Critical thinking question. Why? Why do you think our body gets rid of the skeletal muscle cells if we are not using them? I want you to think like your body. You are a body. Think like one. Why does the body get rid of the skeletal muscle cell?
Energy, like conserving energy.
Absolutely. Absolutely. A skeletal muscle cell is very metabolically expensive to maintain. It's too much ATP. It requires too much material to keep it up. So if we are not using it, our body's going to say, hey, why am I carrying all of this muscle around? I'm going to get rid of some of this muscle to help conserve energy for my body. Okay? All right, last little video here we're going to watch that talks about how do we grow our muscles.
Muscles. We have over 600 of them. They make up between 1/3 and 1/2 of our body weight. And along with connective tissue, they bind us together, hold us up, and help us move. And whether or not bodybuilding is your hobby, muscles need your constant attention because the way you treat them on a daily basis determines whether they will wither or grow. Say you're standing in front of a door, ready to pull it open. Your brain and muscles are perfectly poised to help you achieve this goal. First, your brain sends a signal to motor neurons inside your arm. When they receive this message, they fire, causing muscles to contract and relax, which pull on the bones in your arm and generate the needed movement. The bigger the challenge becomes, the bigger the brain's signal grows, and the more motor units it rallies to help you achieve your task. But what if the door is made of solid iron? At this point, your arm muscles alone won't be able to generate enough tension to pull it open, so your brain appeals to other muscles for help. You plant your feet, tighten your belly, and tense your back, generating enough force to yank it open. Your nervous system has just leveraged the resources you already have, other muscles, to meet the demand. While all this is happening, your muscle fibers undergo another kind of cellular change. As you expose them to stress, they experience microscopic damage, which in this context is a good thing. In response, the injured cells release inflammatory molecules called cytokines that activate the immune system to repair the injury. This is when the muscle-building magic happens. The greater the damage to the muscle tissue, the more your body will need to repair itself. The resulting cycle of damage and repair eventually makes muscles bigger and stronger as they adapt to progressively greater demands. Since our bodies have already adapted to most everyday activities, Those generally don't produce enough stress to stimulate new muscle growth. So, to build new muscle, a process called hypertrophy, our cells need to be exposed to higher workloads than they are used to. In fact, if you don't continuously expose your muscles to some resistance, they will shrink, a process known as muscular atrophy. In contrast, exposing the muscle to a high degree of tension, especially while the muscle is lengthening, also called an eccentric contraction, generates effective conditions for new growth. However, muscles rely on more than just activity to grow. Without proper nutrition, hormones, and rest, your body would never be able to repair damaged muscle fibers. Protein in our diet preserves muscle mass by providing the building blocks for new tissue in the form of amino acids. Adequate protein intake, along with naturally occurring hormones like insulin-like growth factor and testosterone, help shift the body into a state where tissue is repaired and grown. This vital repair process mainly occurs when we're resting, especially at night while sleeping. Gender and age affect this repair mechanism, which is why young men with more testosterone have a leg up in the muscle building game. Genetic factors also play a role in one's ability to grow muscle. Some people have more robust immune reactions to muscle damage and are better able to repair and replace damaged muscle fibers, increasing their muscle building potential. The body responds to the demands you place on it. If you tear your muscles up, eat right, rest, and repeat, you'll create the conditions to make your muscles as big and strong as possible. It is with muscles as it is with life. Meaningful growth requires challenge and stress.
All right, folks, now you know how to grow your muscles. Any questions, guys? All right, so we're going to stop right here, and we are going to continue this on Thursday and finish this chapter then. Have a great day, folks. See you then. Thank you.